Hey, this is JC on a bike. Thanks for joining me. On this ride, I'm going to talk about the new Nature Medicine paper, the Proximal Origins of the SARS-CoV2 virus, and I'm also going to mention this other Nature paper which recently came out by one of those same authors showing the various recently discovered coronavirus variants that are found in pangolins, specifically pangolins that were sort of stopped by customs in China in 2017, 18, and 19. So very interesting data. Join me on the bike. Good afternoon, Pittsburgh and universe. This is JC on a bike. My name is JC and this is Journal Club on a Bike. I'm coming to you from a lockdown Pittsburgh again. We're going to do the Nature Medicine paper that recently came out titled The Proximal Origins of SARS-CoV2. In this paper they discuss various possible origins of the current pandemic and address the sort of mythology that is being whipped up from all sides regarding the natural versus laboratory origin stories that, quite frankly, are both capable of explaining the existence of this virus. The biology of this virus can be explained in multiple ways. And this paper concludes at the end that the most parsimonious explanation is a zoonotic emergence, either through an intermediate animal from bats to humans or by human to human within human selection against the ACE2 receptor to enrich the spike protein to produce the novel affinity that we see in the sequence in SARS-CoV2. So, I mean, the long and the short of it is that this Nature paper puts forward the same argument that Michael Olsterholm put forward on Joe Rogan's podcast, and puts forward the same argument that I've heard from a number of other sources, that we would have never come up with this sequence, therefore it's not from a lab. This is exactly the same argument. There's no difference at all, even though this paper uses a lot more words. They're talking about all the same things that, that independent bloggers have been talking about for a while. So first they talked about the binding domain, the receptor binding domain of the spike protein. Just like we have talked about it, what's special about it is that it is an ACE2 targeting spike protein, and that what's interesting about it, they say, is that spike protein could not have originated in a laboratory because it's such a unique and unpredictably strong affinity for the ACE2 receptor. And they discuss this in the first, few paragraphs of this review. Then they go on to talk about the furin proteases, the furin protease site, and they say that they don't know what that does on the SARS-CoV virus, but they know what it does on a lot of other viruses. The cleavage or partial cleavage of the S1, S2 domains of spike proteins facilitates cell entry. And if you want to do your own research on that, you can. There's hundreds of papers that talk about using trypsin cleavage. They use lots of different enzymes to show that partial cleaving of the spike protein is what facilitates cell entry. And again, I'm not a virologist, but these things are not hidden in the primary literature. And we came across these kinds of things months ago already. We knew this already months ago. So these molecular biologists aren't telling us anything we didn't know. and not telling us anything we didn't talk about on earlier rides. They're just getting more specific about it. So then they also talk about the idea that there are some glycosylation sites on the virus, and these glycosylation sites can sort of hide epitopes on the virus and aid in invasion. And they cite a paper there which is a review about glycosylation of viruses and the role that glycosylation plays. So what that basically is that certain proteins, certain sequences will have a tendency to be modified post-translation. And one of the ways that proteins can be modified post-translation is that you can add sugar groups to certain places. That's what glycosylation is basically. I mean, I'm sure there are biochemists in the world that are going, oh my God, JC on a bike's an idiot. But let's just say for the broad audience that wants to know what's going on with this frickin' COVID-19 bullshit, then saying that is enough. And they understand that because they've done crystallization studies on all these viruses before. And I've shown you those before. So what I find interesting about this paper then, after they talk about these three basic, unique things about SARS-CoV2 and how it compares to the other SARS viruses, then they go down the line in this paper and they list the three likely scenarios, right? The first one is in a lab through their first citation really is the receptor binding domain is so unique that there's no way that we would have designed it or done it in a laboratory. So that's one thing they say again in this little summary thing. And they could beat that dead horse all they want. It doesn't make it true. And what they do talk about, though, what I find very interesting is they do mention a couple studies where they enrich avian flu on a background of chickens. And they mention that, we have a lot of receptor homology with ferrets and cats in the bottom of the first column. But then they go on to completely ignore the fact that we've actually used ferrets as an enrichment host to great success with avian flu. And we've even created a respiratory version of avian flu that infects ferrets, and it can be pretty well assumed, although it hasn't been tested, because ferrets have such a receptor homology with us in terms of ACE2, as they say in this very paper, then the experiments that were done in 2011 in the Netherlands and at the University of Wisconsin, these are part and parcel for exactly what they're talking about here, but they purposefully don't include it in the bibliography. They just leave them out. And why do they leave those papers out? Those papers are extremely relevant, and they're more relevant than the chick paper, that's number 17 that they cite, where they're enriching in chickens. It's more relevant than that because the point you make in the first couple columns is that the only way you could really do it is in an animal that had sufficient homology with human receptors, and you list them as ferrets, cats, and whatever. But then when you cite enrichment papers that are using these techniques when you cite those papers, or the papers that you choose to cite, completely ignore the most powerful the most powerful examples of those. The most powerful examples of those are the paper by the Faucier group at Erasmus in Rotterdam and this Japanese fella that works at the University of Wisconsin-Madison, or at least did in 2011. Then they go on to say something very interesting that I find very, very hard to understand molecularly, that they say that Furthermore, it's probably not a chimera, a laboratory recombined virus, because then they would have probably, and they keep using the word probably and could and whatever, they would have probably or most likely used one of the known reverse genetic tools that we have for RNA viruses, for coronaviruses. Now that right there is an incredibly revealing statement, right? Because if you want to be conspiracy theory about it, I mean, if you thought, for example, that this is going to be a disaster. If you thought that there was any way that you could use sort of genetic techniques. Can I sneak by you? Thanks. Can I get by you? Thank you. You can use genetic techniques to make coronaviruses in the way that they did, for example, in the 2015 paper, by taking pieces of one and putting it on a piece of another, or the backbone of another. There was never another paper like that. There's a real easy reason why, because can I sneak past you? Thank you. No talking. We don't want to say hi to anybody. We're going to go to hell in a handbasket, I'm telling you. Thank you. But they don't cover those two papers. They leave them out. And so, oh yeah, that's where I was going. So then the second point they make in their summary little statement on the first page on the right is that they make this statement that they would have used these, known genetic tools, reverse genetics tools, and they don't see any known backbone, so that's without a doubt. Now, if you were going to make bioweapons, or you were going to screw around with gain of function, but you didn't want anybody to know about it, you just wouldn't publish the genetic tools, right? I mean, it's ridiculous. And I'm going to flash a paper up right now from, I think it's 2015 also, which shows that, they were able to already very quickly adapt CRISPR-Cas9 techniques to these viruses. And it was pretty trivial to do so. So the idea that somewhere in the literature, or somewhere buried in a laboratory, or somewhere in classified virus research, that there's not a set of molecular techniques that can explain how this virus could have been assembled from a bat backbone and a pangolin spike protein, you're just being absurd. And again, you're being obtuse. And that's again why they don't include that paper in the frickin' bibliography. Perhaps the most relevant paper of all is not in the bibliography. So there's 3 papers so far that we covered, 4 papers that we've covered so far that they don't even talk about. And these are like, seminal papers in terms of our evolving understanding of how coronaviruses work and how we can manipulate them. So this paper also conveniently, kind of glosses over all the things we can do with culture systems and tries to use as few words as possible to talk about selection through passage. But, I've shown papers where we have lung epithelial cell culture techniques so that we can select viruses on the background of a culture of lung epithelium. So we have culture techniques that we've developed which could be used to select for variants in pangolins, variants in bats, and then find those useful variants and then take them and use known or unknown genetic tools to snip pieces of those off. And you can't tell me that you wouldn't know that. We've determined already in many of these papers, the papers are specifically discussing the functional properties of the different proteins in these viruses. So it's not like we don't know and we haven't looked into the function of these different proteins. And so swapping them between viruses is not so absurd because we have the molecular techniques for it. And so once again, just let me make very clear, I'm not at all suggesting that I know definitively the origins of this virus. I'm not telling you I understand what we're up against. But I am definitely saying, I am claiming that people are lying to us when they say that we couldn't have made it, that we don't know of any viral labs in the world that would be doing research that might be considered similar to this virus generating or finding or cultivating a virus like this. It's just absolutely absurd. And so then out comes this paper in Nature a couple weeks ago, or even a couple days ago, where they show extreme homology between pangolins coronaviruses and bat coronaviruses and the human CoV2. This paper is, in my humble opinion, a big giant smokescreen because they don't really do anything in the paper. They show you cartoons in figure 1 that show you that the rough functional arrangement of the viruses in the pangolin is very similar to SARS-CoV2, but no more similar than the bat. The bat is still more similar. So it's not clear to me what they're implying here. Do they think that a bat and a pangolin mixed together in the wet market? Like, that's kind of absurd. The other thing that is interesting about this paper is that they're using a consensus sequence. So they looked at like 43 weird samples taken from pangolins 2 years ago. It's from 18 pangolins. 43 samples were analyzed, 6 were positive for something, which they sequenced and claimed that there were 6 different coronavirus sequences. And then they took 2 of those and made a consensus sequence. And then they tested other samples using PCR based on those viruses they analyzed from the 6 samples that were positive. So what does that really mean? I don't even know. So you used primers you designed from viruses you claim to have found in pangolins from 2 years ago or 3 years ago, and you used those primers to test pangolins from March of 2019, and they also tested positive, and I can't tell for sure if they isolated the virus from 2019 or not, because they're always using a consensus sequence, like a mix between the pangolins from 2018 and the pangolins from collected in 2019, I think. And so then when they compared in figure 2, interestingly enough, the highest homology is with the bat. You can see that, I think, in green. And then right around the spike protein, the homology just crashes off. And instead, the homology rises with the pangolin sequence. And so what it looks like to me, although, you can ask other people and tell people, see if people say I'm full of baloney, but it looked like to me what this figure indicates is that the easiest way to get here is not through a series of evolutionary randomness from pangolin to bat or from bat to pangolin and then to human. The simplest way to do it would be one step. Take the spike protein from the pangolin and put it on that bat. Done. And so to me, these 2 papers combine to give you all the puzzle pieces you need to say that we could have easily made this virus. Never mind the possibility of taking a pangolin bad virus chimera and then growing that chimeric virus in a ferret colony, because then you would just get far enough away from the original clade by the time it passed 3 or 4 times and enriched itself a little better. You'd have a novel coronavirus that didn't look like a chimera at all, because you passed it through ferrets a few times and allowed the natural mutation rate to hide your trick. Not saying it happened. Hey guys, thanks for joining me on this ride. This ride is about the new Nature Medicine paper that came out a few days ago entitled The Proximal Origins of SARS-CoV2. It's an interesting paper that actually puts forth a number of plausible scenarios wherein this virus could have been created in a laboratory, but then dismisses them as not the most parsimonious explanation for the origin of the virus, and instead come up with some kind of combination of bats and pangolins, or a bat jump which then evolved in a smaller human population before becoming the pandemic potential that it's now showing. I'm not going to suggest to you that I can argue with any of these people because I'm not a professional virologist, but I have been reading for a number of weeks now, and it leads me to come to the conclusion that there's not really much said in this paper. Number one, they list many of the different ways which we have suggested before, but more important to understanding the motivation behind this paper, look at the examples that they give you for the specific laboratory methodologies that they then discount. For example, they talk about the enrichment of avian flu, specifically in chickens. This is really interesting because in the paragraph before that, they talk about the only way that this virus's receptor binding domain could have been enriched in the way that it appears to have been enriched is through selection in a human or human-like host, like ferrets or cats. And then in the next paragraph, they talk about enrichment in hosts, but then they specifically omit the fact that we've actually used ferrets for real to enrich avian flu in 2011 at the University of Wisconsin and in the Netherlands, and we got a great gain of function which resulted in an aerosolized flu virus. And this was lauded as the most awful experiment that had been done in virology, and a lot of people condemned it. In fact, the author of that paper had to fight for about 1.5 years to get the partial manuscript published, and then ever after that, the Dutch government ruled that his line of research was illegal. They just didn't fund it anymore. So interestingly, his original research was also funded by the American NIH, and I'm not sure if it was a combination of Dutch money and American money, but for sure American money was involved. So again, what I'm pointing out is that if you write an opinion piece like this and then leave out very key pieces which are actually more related to the point that you're trying to discount, it's kind of frighteningly obvious that you're being obtuse. So there's a couple more statements that they make in this paper which I find shocking. The first one is that they say they say that the known genetic tools, the reverse genetics tools that are known to work on coronaviruses were not used because they don't find any backbone signatures, any known backbones in the sequence that they have. And this is just mind-blowing because what it suggests to you then, they actually say the words probably would have used known tools, suggests to me that there are tools that we don't know about, and they probably don't have to then, right? Probably gives a pretty big hole for other options. And so with each one of these laboratory options, including passage through cell lines, they give you a very cursory example of why it couldn't have worked, while ignoring the mountain of examples in the primary literature of where it has worked in the past with other viruses and other types of viruses, even beyond coronaviruses and influenza viruses. So join me for the ride. I think it's a really great discussion that tries to break this down. I might even use this for the wrap because this was actually more detailed than I wanted to go. But anyway, enjoy me for the ride. I think it's really important. It's really great to try and be able to provide this information for you guys. I'm finding it a lot of fun. It's a way for me to also process it in my own head. And so I appreciate you guys watching. Thanks, and I'll see you next time.